Is having a strong academic background, including PhD in botany from Madras Hindu University. His research focused on improving seed longevity and disease resistance in soybean. Currently, Dr. Kuldeep sir is leading and collaborating on multiple research projects, including the development of a rapid detection kit for seed longevity and pathogen identification. His contributions include groundbreaking discoveries in seed viability assessment, genetic markers for allium species, and virus-free garlic seed production. Sir is a recipient of numerous awards and fellowships. Dr. Kuldeep sir has made a significant strides in agriculture biotechnology and published around 40 research papers in reputed national and international journals with high impact and mass scope. His work continues to advance the seed quality improvement and molecular breeding for sustainable agriculture. In this session, sir will deliver a talk on plant immunogenic memory and seed-mediated immunogenic memory transfer. Sir, over to you, Kuldeep sir. You can proceed your presentation.
Thank you, sir. I'm very thankful to Dr. Manjan Gowda for giving me such a great opportunity and introducing me to all of you. I'm very thankful to all of you who are joining this presentation today. And I'm also very thankful to all my seniors, Dr. Ramya Madam, Dr. Naik sir, and other my seniors. Really, it is great privilege and honor to me to deliver my lecture on how immunogenic memory play a key role in seed allium's immunity. So, dear audience, sir and madam, as you are well aware that in animal system, we have well-developed immune system, B cell and T cell, and they produce the antibody, and we can cope up various type of pathogen. When new strain come, new pathogen infect us, our body by homologous recombination produce new type of B cell and T cell so that we can cope up with various type of pathogen. But in case of plant system, that immune system is not as much complex as mammalian's immunity. The system is less complex and they lack immune cell like B cell, T cell, and other circulatory system. But still, plant immune response is highly specific and lifelong memory which can transfer from one generation to other generation and also restrict to self-reactivity. So question arises, how does plant immune system do this? So they follow different strategy. As we are aware that when pathogen infect the plants, they release some molecule like PAMP, pathogen associated molecular pattern. And this is recognized by the plant's plasma membrane receptors known as pattern recognition receptor. When pathogen PAMP recognized by the PRR and signaling occur, they lead to the resistance. But sometimes when PRR not recognize the PAMP and this lead to the susceptibility, or in some cases PAMP, pathogen PAMP like polysaccharide, chitin, other molecules are recognized by the plant's pattern recognition receptor but signaling not occur, downstream signaling not occur, so it lead to the susceptibility. Later on, when PAMP recognized by the PRR and then it lead to the cultivar specific resistance. So there are evolutionary relationship like you can look in my slide that PAMPES, pathogen associated molecular pattern, means polysaccharide, chitin, lipids, some molecules act as a pathogen released by the pathogen, and plants develop immunity against its PAMP triggered immunity. Then what pathogen do? They develop new type of effectors and new type of toxic molecule. When they release this again in the plants, then plants become susceptible because pathogen evolve and develop some new type of effectors known as effector triggered susceptibility. When plants are very smart, in nature, lots of crossing are occurring, and during domestication, we lose from wild allium to cultivated allium some important genes like disease resistance. But sometimes plants evolve and they develop effector triggered immunity against that new strain. Then vice versa again, pathogen release some molecules could lead to susceptibility, and then again plants develop immunity against new strains. Like that, pathogen evolve and then plants evolve. So primary defense in plants mostly occur due to plant cell wall, like pathogen-associated molecular pattern are AMP, MAMP, like lipopolysaccharide, peptidoglycan, and bacterial flagellin. And these uh, pathogen molecules rejected by the plasma membrane of the plants, pattern recognition receptor, and they lead to the plant immunity, like flagellin sensitive to in Thaliana toll-like receptor in 5 in human. Like if pathogen release some effector, so flagellin sensitive to in Thaliana toll-like receptor is similar to the human and lead to the PAMP triggered immunity. Like that, plants evolve various mechanisms like chitinase and enzyme, which lead to the tolerance. Ross et al. Proved that when they infected the tobacco plant with these viruses, TMD, tobacco mosaic virus infection, plant develop some immunity when we infect it by other pathogen. And they proved that plants have memory up to the 20 days and memory lasts after 20 days. Like we have the vaccine of the COVID and first, second, third vaccination, we develop immunity and memory. Plants also have some memory due to the systematic acquired resistance. And earlier, it is well known that gene model work well. One gene, one R gene recognize one strain or one pathogen and no somatic DNA arrangement similar to immunoglobulin. So, but question arise, single R gene recognize single effector, but in nature, lots of effector are I discussed earlier. Pathogen are very smart. They are releasing many type of effector and plants have only 50, 60 or sometimes 200 to 500 R genes. So how 500 R gene can recognize such a large number of pathogen? Because no somatic rearrangement similar to immunoglobulin in plants. So how plant recognize diverse effector? So it's a very tough question, but scientists answer to this question. Mostly plant are gene present in gene cluster where higher rate of recombination occur. So if a new type of pathogen comes, so there is guard hypothesis where RIN4 is a common target. Like if we capture the director of the ICR Institute, we can capture all the scientists like RIN4. If RIN4 is captured by the pathogen, then mostly plant will be destroyed. Plant will be destroyed by the pathogen. But if RIN4 is a master regulator, if RIN4 identify the all type of effectors and they do the downstream signaling and then lead to the PAMP triggered immunity or effector triggered immunity. As you can see in the unchallenged plant, the RIN4 is in control stage and it is not phosphorylated by the RIPK. And when a pathogen is not infecting the plants, the R gene like RPM1 and RPS2 are in inactive stage. But dear sir and madam and audience, if pathogen infect the plants, then RIPK phosphorylate the RIN4. When RIN4 is phosphorylated, it do the conformational changes in the RPM1 and RPS2, and due to phosphorylation of the RPM1, it lead to the activation of the R protein antimicrobial compound, and this lead to the effector triggered immunity. If some new type of pathogen enter new type of effector, then RIN4 is cleaved and then RPS2 phosphorylate and it activates the RG. So dear sir and madam, you know, RIN4 is a basic target which recognize most of the pathogen and lead to the downstream signaling, mostly RGs. So why RIN4 is favorable target? Because RIN4 interact with H plus ATPase. Because of this gradient, RIN4 play important role in stomata opening. If stomata is closed, then pathogen could not enter. If stomata is open, pathogen could enter. So well-known role of RIN4 is not known, the mechanism is not known, but it is known that RIN4 acts as a guardy protein and it totally play important role to downstream signaling. And by closing the stomata, they prevent the infestation of the pathogen. Further, RIN4 activate various R genes, and sometimes due to systematic acquired resistance, plant develops some apoptosis or cell death, and this lead to the caspase. So prevent the spreading of the disease and lead to the immunity. So how R gene work actually? Sometimes we cross the wild plant with the cultivated one. Even sometimes within cultivated one, the pollination is not successful. This occurs due to hybrid necrosis. Scientists mapped it. So they reported some R gene activated and they consider the other plant pollen grain as a foreign molecules. So sometimes it is also known that circadian rhythm, day and night infection also lead to the plant immunity. So overall, the animal immunity have the mobile immune cell and antibody, but plant immune signaling occur due to the salicylic acid, jasmonic acid, and other molecule. As in this diagram, we are showing you that when pathogen infect the plant, azelaic acid, methyl salicylic acid, jasmonic acid, glycerol 3-phosphate like mobile immune signal and lipid transfer protein move from one places to other places by xylem and phloem and lead to the systematic acquired resistance. Further, salicylic acid lead to the secretion of PR proteins and this lead to the antimicrobial activity. Overall, epigenomic changes also play crucial role for development of the resistance like salicylic acid, which lead to the chromatin modification and priming of the DNA by methylation and acetylation, and sometimes salicylic acid also lead to the homologous recombination with the help of R1851 and BRCA2 similar to the human being. So this lead to the transgenerational immune memory. So in plant with salicylic acid, jasmonic acid, or any BTH and along, so this lead to the activation of the MAP kinase 6, and MAP kinase 6 accumulate in the cell, and when the infection comes, they amplify the signaling cascade and activate the R gene and NPR and have long-lasting memory. So transgenerational memory and immunity occur due to the R gene cluster, and there are some homologous recombination, epigenetic modification lead to the plant immunity. But there is a need to study immune function and memory operate in large population and need to study immune function in crop like tea and alliums. So here I want to show you a case study. Like in tea, transcriptome analysis reveal candidate genes involved in blister blight defense in tea, Camellia sinensis. So what we did in this study, we have taken the two plants, SA6 resistance and Kangra asa susceptible, and we collected sample at 4 stages. 4 stages, 2 plants with 8 sample. When we inoculate the spore, and then spore might germinate at 7th day, and 3rd, episoria development and mycelium development, and then 4th, sporulation and secondary infection. 8 sample and 4 each of resistant and susceptible source. We did the transcriptome sequencing of SA6 resistance and Kangra asa. And the scan electron microscopy clearly revealed that at left side resistant plant is there, no infection, and right hand side you can look the infection of the Ixobagidium vecgens, which lead to the blister blight disease in the tea. And when we did the sequencing and we mined the data, so we find the GO, K is easy, EC, and then we did the differential gene DEG, differential expression of the gene. So few of the genes identified in this study, but we was more interested to identify the R gene or immune related genes. So here we identified around 36 R genes, which mostly belong to the RPM1 and RPS2, and around 31 defense enzyme transcript, then metacapsid and chapran and other working non-transcription factor, retrotransposons and transporters. Dear student madam, what happened when pathogen infects the plant? Rim-4 protein activate, and the RPM1 and RPS2 activated by these working and non-transcription factor would bind to the upstream of the R gene, and this lead to the activation of the defense enzyme like SOD and other molecule like chapran, which stabilize the expression of the R gene, and LTR copia is transposons, which move from one place to other place in genome, like jumping genes, Barbara Mankin-Clark, who awarded the Nobel Prize. So these transposons move from one place to other place, might be in R gene, so produce new type of R genes so that they can cope up with the new variant of the pathogen. So most of the R gene defense TF, transposons, and MDR are highly expressed in resistance plant at fourth stage. You can see at red color is highly expressed and green color is less expressed. So heat map reveal that R gene in resistance source at fourth stage, they have higher expression of the RPM1, RPS2 like R genes and defense enzyme like acetyltransferase and peroxidase, carboxysterase, and other working and NAM transition factors are highly expressed in resistance source compared to the susceptible, and retrotransposons are highly expressed in SA6 at fourth stage. Different transporters who detoxify the toxic compound produced by the exobacterium genes also detoxify here, and defense regulatory genes are highly expressed in resistance source also. So most of defense regulatory genes like Chaptron and cytochrome P450 metacapsules play a very important role in defense. Chaptron actually stabilize the R protein, and cytochrome P450 detoxify the cell. Metacapsules play important role in cell death. Circadian rhythm play important role to prevent the disease. Multi-drug resistant transporter, mostly they remove the toxic compound from the cell. LTR copia move from one place to other place in genome, so create new type of R gene so that plant can tolerate to infestation of the pathogen. But dear sir and madam, we identified more than 140 genes which might play very important role in disease resistance and immunity, but which gene is very important for which we can go for gain of function and loss of function. My means, if you say these genes are playing very important role, until and unless you will do the gain of function and loss of function, you could not say that this gene is working. My mean, if you remove the some gene from susceptible plant, they become the resistance. Then we can say, okay, this gene is working and this gene is responsible. If you clone some R gene from resistance source, 1 or 2 or 3 genes, and transfer it to the susceptible plant, and susceptible plant become the resistant, then we can say, okay, this gene is working. So that's why from 146 genes, we have to reach 1 or 2 genes which might play important role in disease resistance. So for that, we did the protein-protein interaction analysis, and we identified that among them, few of the highlighted R genes, defense enzyme, transcription factor, MDR, they are controlling the other genes. Like if we hijack the director of the institute, we can control all the scientists like that. Among 140 genes, these 5-6 genes of the R gene, which is highlighted here in the map, they are playing key roles. So protein-protein interaction could be a good option to identify or sort down the gene. Then we can put the real-time PCR and can check, okay, really these genes are working or not. So we prove that RNA sequencing data with RT-PCR data, there is a coherence between RNA sequencing data and real-time PCR data. So we hypothesize in this study that when pathogen come in resistant plant, RIN4 activate and phosphorylated by the RIKP, and then it activates the RPM1 and activate the R gene. Further, various Purki and NAT transcription factor which bind to the upstream of the RIN4 and R genes activate these R gene and pathogenesis related protein. The transposons like LTR move from one place to other place in R gene, so create new type of R gene and lead to the transgenerational immune memory. For the transporter, which detoxify the cell toxic compound, which is released by the E genes, play important role in immunity. And very important, SOD, another square, the free radicals, and MAP kinase 6 when secondary infection occur, so they accumulate, and when real secondary infection, in primary infection, MAP kinase 6 accumulate, and when secondary infection come, they amplify the signaling cascade and lead to the high immunity against the blister blight defense. So this is the story of the T. Now we will move ahead in the story of the LEMs. So marker-restricted identification of LEM genotypes containing immunogenic memory against anthracnose. Dear sir and madam, as you know that allium cepa and allium sativum, pyaz and lasun are major commodity being cultivated in a large scale, but due to the climate change, other factor, the production are reducing. And during evolution from wild allium to cultivated allium, these cultivated allium lose some of the biotic and abiotic tolerance trait. Among them, like anthracnose, stem blight, purple blast disease, and various viruses which infect the alliums and lead to the 100% yield loss. So now there's a need of our to screen those wild germplasm which have resistance to cope up the biotic and abiotic stress and identify the genes and trait and introduce these genes and trait into the elite cultivar of the allium cepa. But if you are not doing this and using agrochemicals, which is hazardous to human being and food chain also, there is a need to identify those germplasm and the genes which is responsible for disease resistance, immunity, and memory. So it is very important to identify and diagnose resistance adaptive traits in wild germplasm and introduce it into the elite cultivar. Besides this agrochemical approach and molecular breeding approach, it is very important to identify the immunity or memory like genes. Just we have the vaccination and we develop the immunity. So like that, plants might have some immune memory and we can identify this in wild allium and can introduce into the cultivated allium. Some of the crops where pathogen attack the plants, they develop the hypersensitive response and immunity against the specific pathogen and lead to the systematic acquired resistance as I discussed by moving the azelaic salicylic jasmonic acid by xylem and phloem. So repeated pathogen infestation lead to the accumulation of the MAP kinase like signaling molecule, and when the pathogen really infect the plants, they can cope up well because MAP kinase is already accumulated in the plant, and when real pathogen come, they amplify the signaling cascade and lead to the plant's immunity. But these immunity is highly specific and transgenerational immune memory and provide resistance against broad range of pathogen and in fact biotic abiotic stresses. It means if plants have the memory and immunomodulated by some immunomodulator, the immune memory might be transferred 1 or 2 generations through the seed in next generation. So I hope that immunomodulator and pathogen infestation lead to the high stress tolerance, gene expression, deposition of callose like molecule and catenase metabolite in vascular bundle, and upregulated expression of the immune coding gene. They might close the stomata, deposit the bags, and maintain the intact epidermis and vascular bundle in the plants. So, dear sir and madam and audience, to understand the immunogenic memory in the plants, and it is essential to identify the wild allium subset because I had around 96 wild allium, and for all the plants I could not screen because it is very costly to screen. Hello, who are you speaking?
Yes, sir. Yes, sir. You continue, sir. No issue.
Okay. So to understand the immunological memory, it is essential to identify the wild allium subset because in 96 wild allium, I could not do the experiment. So subset is very important. From 96, we had to reach 23 or 25. So for making the subset from 96 to 23, it is very important to make the subset by using molecular markers. Because if you use the molecular markers, it will eliminate the duplicate sample, maximize the genetic diversity, minimize the time and expenses, and it could be possible by developing a desired wild allium subset from the diverse wild allium subset. So dear sir and madam, ILP markers, intron length polymorphic marker, is novel marker for understanding the diversification and selection of subset of wild allium from 96 wild allium, and then from 23 wild allium to decipher the relationship of 23 wild allium with 2 cultivated allium. We need chloroplast marker which are superior to ILP to reveal the evolutionary relationship and kinship. Hence, CPSSR and chloroplast SSR and ILP marker genotyping are desirable for identifying the contrasting wild allium or studying the allium's immunity. But unfortunately, the development of subset of wild allium and the mechanism of action and effect of repeated application of immunomodulator to develop immune memory for combating the effect of disease in allium are still lacking. So that's why which is immensely required for improving the yields of allium. So I hypothesize, particularly in this study, that ILP markers might able to give me the subset or mini core collection, and chloroplast marker might be able to reveal the kinship of wild allium with cultivated allium. Second hypothesis, immunomodulator like salicylic acid, jasmonic acid, and C. pilospidis infestation to the wild allium in relation to the cultivated allium may lead to the development of immunogenic memory against antagonists by depositing pre-stress molecule like PAP kinase, may exhibit high stress tolerance gene expression, may deposit callose catenase like metabolites, may close the stomatotropism for high expression, deposit the wax, maintain the intact epidermis and muscular bundle, and upregulate the immune coding genes. And wild allium putative immune memory genes may help in combating the anthracnose disease. So we have the four objectives here: to develop the allium CPA and allium sativum ILP markers, then develop and validate chloroplast marker of wild allium and cultivated allium, then immunomodulator and marker-assisted identification of allium genotypes containing immunogenic memory against anthracnose, and then identification of putative allium's immunity genes by transcriptome. So, dear sir and madam, as far as objective 1 is concerned, ILP marker development in allium CPA, we mined around 20,000 EST and developed more than 2,000 markers and around 500 markers screen other 10 allium CPA individuals, and they are polymorphic and cross-transferable. They can distinguish the allium sativum and wild allium and cultivated allium in 3 groups, and they are highly cross-transferable, means allium CPA markers are working in wild allium and cultivated allium sativum. It means if we want to reduce wild allium genes into cultivated one, these markers could be good for marker-assisted integration of the important genes. And then we had the 96 allium CPA population, and we proved that these ILP markers are classifying these cultivated allium CPA in 2 groups: low TSS, low juice of onion, and high juice of onion. So these markers are good. Then population structure also revealed that 96 allium CPA are falling in 2 groups: high TSS, high total soluble content, and low total soluble contents. Then, as I discussed, I developed the allium sativum ILP markers. So we mined more than 20,000 TST and developed more than 1,000 markers and validated 100 markers on 23 individuals, and they are showing mostly polymorphic and cross-transferable. They are also, you can see the polymorphism level in allium CPA, allium sativum, wild allium. Diverse band are there, polymorphic and cross-transferable. And these markers, allium sativum ILP markers, are also clustering allium CPA, wild allium, and allium sativum in 3 groups. And when we validated these markers on 52 population of the allium sativa, short day and long day, they are classified in 2 groups, and population structure are also classifying it in 2 groups. So in allium CPA and allium sativum ILP markers, there are morphological and genotyping consistency in grouping. So we can say that these ILP markers are suitable to develop the sub-core collection or subsample of a wild allium. Then, dear sir and madam, second objective is to develop the chloroplast SSR marker from wild allium and cultivated allium. So for allium paradoxum wild allium, we downloaded the whole genome of the chloroplast and developed 15 allium paradoxum chloroplast marker and validated it. So you can see the polymorphism. The bands are distinguishing from each other, and they also classify the wild allium in 2 groups. So it is good. Then allium CPA and allium sativum chloroplast marker we have developed. Around 22 chloroplast marker we have developed, and they also showing the polymorphism and cross-transferable, and they also classify wild allium, allium sativum, and allium CPA in 3 groups. So we can say these chloroplast markers are also good, and then we validated this and 79 wild allium. So wild allium are two types, mostly flat leaves like garlic leaf, lasun leaf, and fistular fistular waxy leaf like onion type round leaves. So they are classifying into group flat and fistular. So we can say these markers are good. So overall now for me the ILP markers is ready to develop the sub-sample from the wild allium, and chloroplast marker are ready to develop to know the evolutionary relationship or kinship. So that's why my third objective is immunomodulator and marker assisted identification of allium genotypes containing immunogenic memory against infect nodes. So what we did here, we screened the 96 wild allium by using ILP marker, and we developed a 23 subsample of the wild allium, and these 23 subsample were genotyped with 22 chloroplast marker with cultivated alliums. So to know the relationship of wild allium with cultivated allium, because we have to select the distantly related wild and cultivated allium, but they must be crossable, and the wild allium might have the immune memory gene. Then these 25 sets of wild allium and cultivated one divided in 3 groups and given the immunomodulator, and then RT-PCR another method, we identify the immune memory response, and then we selected few wild allium and spray the immunomodulator and pathogen, and by transcriptome another approach, we identify few genes which are responsible for the plant immune memory. So as far as immunomodulator and marker identification of alien genotype containing immunogenic against infect nodes objective is concerned, so first we have to identify the subsample of wild allium. So this is the phenotypic data where two type of plants are there, fistular leaf and flat leaves. So based on the leaf, we are classifying this in 2 groups, and based on the genotypic data of the ILP markers, we are classifying it in 2 groups, fistular and flat. So there are consistency between the morphological and the genotypic data, and based on this, we selected the 23 individual by using PowerCode software, which remove the duplicate sample and give the unique 23 samples which represent the whole diversity. And then we have taken the 2 cultivated one allium cepa and allium sativum. So these 25 individual were genotyped with chloroplast marker, and you can see that allium cepa is distantly related with the allium pistulosum, and literature report that allium pistulosum might have some resistance gene. So for future study, we have taken the allium cepa and allium pistulosum. Although these 25 individuals, we are grouped in the 3 group A, B, C. A is provided water spray, B is provided 2-day continuous immunomodulator spray, and C is continuous 1 to 7 day immunomodulator spray. As we are hoping that if human being is vaccinated 1, 2, or thrice, so the person who is immunomodulated thrice, they might have more immunity against the COVID. Like that, the wild allium which immunomodulated till 7 day, having the more expression of the SOD, chaperone, and ABC transporter, and cultivated allium have less expression in 7th day, but 2nd day they have some expression. So we can say that the immunomodulation play crucial role, and wild allium having the more expression of the biotic stress gene in 7-day expression. So they might have the immune memory genes. As we can say at C, wild allium have more immunity, and cultivated allium have less immunity genes expression. And then we have selected only 2 individual, as I discussed, allium pistulosum 2 individual and allium cepa. So 2 wild allium, 1 cultivated allium, and they have been divided into the 6 group. First, we spray only water. Second, we spray only immunomodulator 2 day. C, we spray 7-day continuous immunomodulator. D, we spray only pathogen. C, Gliostritis infestation. E, we spray 2-day immunomodulator plus pathogen. E, we spray 7-day immunomodulator plus pathogen. Again, I am explaining. Control, we spray only water. B, we spray 2-day immunomodulator, like 2-day vaccination. C, we spray 7-day immunomodulator, like 7-time vaccination. Then D, only pathogen, only COVID vaccination, COVID infections like in human being, we can suppose. And E, we spray 2-day immunomodulator and then pathogen, like 2-day vaccination and then pathogen. And in F, 2-day immunomodulator spray plus pathogen, like 7-day immunomodulator spray plus pathogen, like 7-day vaccination and then pathogen. So when we saw the response, we came to know that in phenotypic data, the wild allium having the disease, mostly you can see DEF, allium cepa vima super, develop the disease symptoms, but the wild allium are healthy even after infestation of the immunomodulator. They develop the good height and achieve the good growth. And when we did the section cutting of this transverse section of the leaf and saw the by scanning electron microscopy, we observed that the cultivated allium are deteriorating at D, and E and F, the wild allium mostly depositing the callose and might prevent the pathogen entry. Although here I'm not showing, but we did the fluorescent microscopy where we observed that the callose is highly deposited in wild alliums, but cultivated alliums, callose is very less deposit or not deposit, and leaf is deteriorating. When we did the real-time PCR analysis with SOD, Chevron, and ABC transporter, we came to know that in wild allium, first two, you can see E and F, where we spray the immunomodulator and pathogen, they have the higher expression of the SOD, Chevron, ABC transporter. But if we talk about the Vima super cultivated one, they have the less expression of the genes like SOD, Chevron, and ABC transporter. This clearly reveals that wild allium have more immunogenic memory and more response to the immunomodulator compared to the cultivated one. This might happen because during evolution, the cultivated allium lose these immunogenic memory related genes, and wild allium might have more immunogenic memory genes. Now, need of our using this chloroplast and ILP marker, we can introduce it into the cultivated one. So now, for further study, we selected only two plants, one wild allium and one cultivated allium. One is control and one is 7-day immunomodulator spray plus pathogen. So we have two plants, wild allium and cultivated one. We have two treatment, one is control, other is immunomodulator spray plus pathogen, A and F. So dear sir and madam, you can see at scan electron microscopy figure at A wild allium and F wild allium, treated plant have the more wax deposition and stomata is closed. But allium cepa vima super, which is cultivated one, the control and treatment have no difference. Wax is not deposited. In fact, stomata is open in cultivated one, but wild allium has closed stomata. So this might happen due to rim4, and that's why we can say the wild allium have more resistance, more immunity. When we cut the section, wild allium have the intact epidermis, but cultivated allium are deteriorating and their vascular bundle are destroyed. So C. gliosporitis infestation lead to deterioration of the vascular bundle. If we observe the leaf, root, shoot, all other traits, so mostly the wild allium having the significantly higher plant height, root length, dry bed, everything the wild allium have the higher. You can see the wild allium have more length in the root compared to cultivated ones, so they are more climate resilient. So next objective is, once we identify the few plants so which have the immunity, so identification of putative allium immunity gene by transcriptome methodology. So here we use the wild allium and cultivated alliums, and we have the control and treatment. So wild allium cultivated on two plants and control water spray, and in treatment only 7-day immunomodulator spray plus pathogen spray. So we isolated the RNA and sequence it, and then FPKM fragment per kilo base of transcript per million reads. We analyze the differentially expressed gene we identified, and then we develop the heat map. So you can see, dear sir and madam and audience, that in wild allium, 6-acroslosom, around 28,000 genes are upregulated. But in cultivated allium, only 20,000. And in cultivated alliums, 36,000 genes downregulated. But in wild allium, only 16,000. So in each and every case, the wild allium have the high number of genes upregulated and low number of genes downregulated. But I am more interested in the immunity-related gene, like R gene, RIN4, RPM1, RPS2, MAP kinase, WRKI, cytochrome P450, MYB transcription factor, other immune coding genes like chevrons, castases. So that's why these genes are upregulated into the wild allium, and these genes are downregulated into the cultivated allium. As far as this heat map is concerned, you can see A small letter denote to the wild allium. Capital letter AF denote to the cultivated allium. So you can see that cultivated alliums, wild allium having the higher percent of the MAP kinase, and wild allium have the higher percent of the worky, higher percent of the RG, low expression of the susceptibility gene, because susceptibility gene lead to susceptibility, which might not express much in the wild, but highly expressed in the cultivated one. Other immunity gene like chaffron, which is like the R gene, are highly expressed in the wild allium, and activator are highly expressed into the wild allium. But if we talk about cultivated one, you can see, dear sir and madam, that MAP kinase is not accumulated into the cultivated allium. So that's why when infection come, the wild allium have the memory, have the MAP kinase, latent MAP kinase proteins and accumulated. When real infection come, they amplify the signaling cascade and lead to the immunity, like we are vaccinated, so our memory is ready. When real infection come, they can fight faster compared to the unvaccinated person. So like that, in plant, MAP kinase 3 accumulated in the plant in wild, but cultivated one, they are not accumulated. That's the reason. That's why wild allium having the more immunity. During evolution, these traits are loosed to the evolution from wild to cultivated one. Now need of power to introduce into the wild allium genes to the cultivated one. Work is less expressed, R gene are less expressed, susceptibility related gene high expressed. Actually, one sweet genes are there. So what sweet gene do? Sweet gene remove the copper from the xylem and phloem. If copper is not available, then antifungal activity will be loosened. So that's why the susceptibility related gene suite might be a target for genome editing in future to making a resistant plant. So other immunity genes are less expressed like chaffron, activator are less expressed. Cytochrome P450 is to remove the detoxify compound, toxic compound from the cells. So they are highly expressed in the wild allium, but less expressed in the cultivated one. And YB transcription factor would bind to the upstream of the R gene and lead to the immunity, are highly expressed in the wild allium. So most of the genes are highly expressed in the wild allium, and you can see by real-time PCR will prove it that the data which NGS next result sequencing are revealing are similar to the real-time PCR data. So we have the confidence in the data. So overall, summary is that the wild allium having the high immunogenic memory. When pathogen infest the plant, RIN4 activate and MAP kinase accumulate, and then real infection come, the R gene activate and lead to the immunity. Worky and MYB transcription factor bind to the upstream of the R gene and lead to the wild allium immunity. And other factor like chaperone and other stabilize the R genes, and the sodium and other enzyme is given free radical and activator lead to the activation of salicylic acid, and cytochrome P450 detoxify the cell, and susceptibility related genes are downregulated into the wild alliums, and ecological trait like wax deposit in the leaf, so prevent the entry of the pathogen. Lignin and the callose deposit into the wild allium vascular bundle, so it lead to the prevent the infestation of the pathogen, and large surface area fruit lead to the climate resilience. So the domestication syndrome which the allium cepa or cultivated allium has might be improved by introducing these desirable traits and genes from wild allium to cultivated allium. So major finding of this study, we developed more than 2,000 markers of allium cepa and sativum, and validated around more than 2,000 markers in allium. So we developed large set of markers for revealing the kinship and revealing the diversity. And further immunomodulator, applying the immunomodulator lead to systematic acquired resistance, reduce the application of the agrochemicals, and wild alliums having the immune memory, and these markers could be used for genomic assisted breeding, and wild allium subsample could be used for many various superior traits, and immunomodulator could be harnessed for improving the immunity. PT2 genes could be transferred from wild allium to cultivated allium, although gain of function and loss of function is important. If we are talking about sweet gene R gene, so we have to delete it from the wild plant, and if it becomes susceptible, then we can say this gene is responsible. And few of the R gene could be mapped, and so overall I can say that these immunomodulator could be used to make the plant immune, and through seed it can be transferred to next generation, and the next generation can tolerate various abiotic stresses. Thank you so much, sir.
Participants, if you have any questions, please ask.
Thank you. Thank you, Kuldeep sir, for very brief presentation
sir.
on seed-mediated immunogenic memory transfer. Really, sir, it was a wonderful presentation. Sir, we appreciate your time and effort for this sharing valuable information. Now the topic is open for discussion. Any clarifications, questions, doubts from participants, you can raise now. Participants, any questions? Any doubts?
If you have any question, please let me know. If anybody have any simple doubt, any question related to marker, wild alien, immunity, memory, R gene, you can ask.